Okay, our next presenter is Lisa McShane from NCI. Lisa was heavily involved in the previous Omics committee at ACR, at NAS, that I showed you the cover of the report. She wrote the inside of it. Lisa.
Correction, I was actually not on that committee. I did give a lot of testimony to that committee, but the committee wrote the book, not me. A couple of disclaimers. Work for the government, so these are my own views, not necessarily the views of the Cancer Institute. What I'm going to be talking about are several examples that I've encountered over the last several years, trying to evaluate omics predictors to determine whether they're really ready to put into a clinical trial. And so I have kind of a unique perspective on this because, well, many of you know what happens in your own research group and you know what you read in publications. I get to see things at many other different points in their development. So I get to see some of the crash and burn stories. I get to see some of the omics predictors that are presented to us for inclusion in a trial that it turns out really are not ready to be included in a trial. I do a lot of work with journals. I get to see things that don't get published because they're seriously flawed. So it just makes life interesting, and I hopefully can give you a few more perspectives than what you might see in your daily routine. So I'm talking really about omics as defined here by the Institute of Medicine panel, but actually what I'm talking about could apply to any other kind of high-dimensional data, whether it's radiomics or even just massive amounts of data pulled from clinical records. So the goal here, which was already nicely described by the previous two speakers, is to take all of this massive amount of information and develop some kind of what I'll call an omics-based test. So that could be based on some fancy artificial intelligence model. It could be some very simple risk score that's just weighting a bunch of omics measurements into a linear score, and you apply a cut point to it and it tells you something clinically. So, the sky's the limit. It could be anything. But at the end of the day, you want to be able to take that test or that predictor or that classifier and apply it to patient data and determine what the next step clinically should be. Are you going to say on the basis of that test result that a patient should get a certain type of therapy, they shouldn't get any therapy at all, that they actually harbor some kind of disease that they didn't know before? So there's a lot of steps that have to go on even after you've done all the mathematics, and it can get very complex, as Giovanni has shown you, a lot of different ways to do the computations, but at the end of the day, it's a very simple question you want to answer. So I do recommend that you read this book. It really has a wealth of good information, goes through a series of case studies of things that include some of the things you've heard about already in Steve's talk, and it gives some recommendations on how to proceed. So after this report was published, the NCI wanted to help operationalize the principles that were put forth in this book. And we also had about 10 or 15 years worth of experience at that point dealing with omics predictors coming from our own funded investigators. So we put all that together and we came up with a checklist that we published in 2013, which I have to say is kind of a compendium of our experiences over that 10 or 15 year period. And we hope that some of the lessons we learned will be useful for you to hear about when you're thinking about developing these kinds of predictors. So we divided it into 5 domains, and since I don't have a lot of time today, I'm only going to talk about the first 3: specimens, assays, and the model development process. I won't get as technical as Giovanni did. I'm trying to keep it more intuitive for the purposes of this talk. So specimens, when someone comes to us with a protocol and says, I have this classifier, this predictor, and I think it'll do this, and I'd like to put it in a clinical trial and validate it. We have found over the years that we shouldn't just immediately accept anything that is handed to us, because some of them do have some serious issues that occurred in the development process. So this list of things that you see here are questions that we start asking immediately. So we'll ask, for example, do you really have a good idea under what conditions you should be collecting these specimens, processing them, and storing them? Do you know how much of the specimen you're going to require? Do you have an approach to screen for the quality? You want to make sure your DNA is not degraded or your RNA is suitable quality. Those kinds of questions. These all need to be written down in sort of an SOP, because we don't want to launch a big trial collecting a bunch of specimens only to find that none of them are usable for the assay. Other issues are feasibility, and you heard a little bit about this earlier today. If it takes 4 weeks to run the assay, are you going to be using that assay in a patient population where they can afford to wait 4 weeks? So you have to make sure you have the kind of capacity and throughput that you need. So these are two actual examples, and I disguised them by leaving out some details. I was actually involved in oversight of a study where they were collecting a particular type of specimen, and they decided they would accrue 100 specimens, and when they reached 100, they would start running their assays as they then continued on collecting more specimens. Well, the unpleasant surprise was that only 20% of the first 100 specimens were actually usable for the assay. It turned out that these specimens and the type of assay that was going to be applied to them required that the specimens be frozen immediately. You couldn't let them sitting out on a table somewhere. So this, unfortunately, was not discovered until 100 cases had already been collected. Another example, we have some archives of specimens that have been collected maybe on some of our older trials. And this was another example where they were going retrospectively instead of prospectively and wanted to run the assays. Well, it turned out that only 15% of these frozen samples were actually usable for the assay. So that kind of groundwork actually should have been done better before they started to figure out whether this was even going to be feasible. Other kinds of things, assay considerations. People, I think, do not often appreciate how tweaking the assay a little bit can have an enormous impact on the downstream results. And we have seen some real disasters this way. For example, locking down the actual assay procedure. Sometimes it turns out that there are required changes in the assay. And I'll give an example shortly where a manufacturer stopped making one of the reagents. And so one of our grantees was halfway through his study, and the reagent he had been using was no longer available. And I'll show you the results in the next slide. Quality monitoring. Is there a system set up in the lab so that if things start going sour, that they will be able to detect it quickly? Quality criteria to accept or reject assay values. I had one example with a study that we were doing, one of our funded investigators was doing, and they were looking at a genomic predictor. And they had a lockdown assay, lockdown predictor, and they got all the results, produced the results, and they didn't quite meet the mark that they were hoping for in terms of performance of this predictor. They went back and started looking at the data and said, oh, well, here, it seems like if we take out this batch, then it works, okay? But there, of course, that was post hoc. So that's not legitimate. You need to have those kinds of acceptance criteria in place at the start. Obviously, you need to worry about analytical performance. If you have an assay that just swings wildly all over the place, there's not much chance that you're going to have it correlate well with some clinical outcome that you're interested in. I mean, this all seems like no-brainers, but you would be surprised how many times we have people come to us who have not thought about these questions. We knew early on in the days of gene expression, microarrays, batch effects. You heard some people talk about that already today. Always a problem. Okay, so what you're looking there at the top, this was one of the older Affymetrix arrays, looking at the perfect match probe intensities. You can see colored in red and blue there, and there's the purple and green. Those were the different batches of assays that were run. So if you're trying to plug in numbers, let's say you developed your predictor on specimens from the red batch, and now you've got the blue batch, and you want to plug those numbers in. Well, that shift that you see there by the shift in the curves is going to affect the performance of your predictor. Same kind of thing still is a problem with even our next generation sequencing data. You have to be very careful that you standardize and that you're really monitoring for shifts, even after you've tried some standardization. So that bottom left plot there, you see those bands of kind of orange and blue. Those correspond to the run dates. Okay, so there should not be bands of orange and blue in that picture. It should be a nice mixture. This was the example I started to tell you about earlier. This one was one of our grant-funded investigators who was building a predictor of response to a particular therapy. He was using gene expression microarrays. And about halfway through his analysis of his hundreds of specimens, they had to switch extraction method. I think something was not available anymore. The kit changed. I don't remember all the details. But he came to us, he contacted me and said, you know, who are some experts at removing batch effects from gene expression microarray data? And we went out on our network and actually got several people to try and help him. At the end of the day, he just threw up his hands and said, it's done. I mean, we can't do it. What you're seeing there in the picture is that the genes that were key to the predictor just had very different profiles depending on which extraction method that they used. He actually stopped his grant and said, I'm not going to pursue this. These data are not useful to me. And I think he ended up actually giving back part of the money. But he said to me, he said, please make sure people understand this. And he not only allowed me to use this picture in my talks, but encouraged me to use this picture in talks. So again, little details. This stuff is not the fun part of doing research. It's the little bitty details that can sometimes get you. Interesting, even more recent example, the MIND-ACT trial. This was a gene expression signature in breast cancer that was aiming to identify women who had better versus poorer outcome, and this may have some implications for who needed chemotherapy and who didn't. If you read the fine print in that paper, you will see that even they were affected by this kind of issue. So there was a change in the RNA extraction solution that was used for the calculation of their signature that caused a temporary shift in the risk calculation for about a 7-month period. They were not told by the manufacturer that they had changed this solution. So they finally detected it after things weren't looking right after about 7 months. Fortunately, it appears that there weren't that many patients or I guess no patient was undertreated, although there were 28 patients in the study who happened to be accrued during that period who got treatment, who would not have gotten it had there not been this change in the RNA extraction solution. Model development and preliminary evaluation. I'd say one of the biggest problems that we find when people come to us with predictors are that number one, they often can't even tell us what the predictor is. They know that their bioinformatics person, their computational biologist, has done some fancy stuff and came up with a model, and they know that they have this computer software they can plug the numbers into and it spits out a result. Okay, but when we get the protocol sometimes, all that will be in the protocol is we have a signature, and here are the 37 genes that are contained in that signature. They have no clue how you put those together. They don't even know if the software has been, really quality checked, if it's stable, anything like that. So one of the first things we do is say, you have to tell us more about your predictor. Exactly how does it work? And this may mean actually getting computer code or having somebody, an independent party, look at the computer code and say, yeah, this computer code is solid code and it actually exists. And that's number one. The other thing that is the second biggest problem is that often these models, these highly computational models, are what we call overfit. Overfitting happens when you fit a very complex model to very high dimensional data, and you end up fitting to noise in the data. And I'll give you a couple of examples of that. And I would say within the last 6 or 7 years, I have had 4 or 5 instances where it turned out when we did the analysis correctly, the predictive performance of the model essentially disappeared. So needless to say, those did not go forward. Okay, so as I said, overfit is when you're really fitting to the noise in the data. And there are lots of ways to do this. One way, which is what we see most often, unfortunately, is where you build this complex model using your whole data set, and then you take the data and you use exactly that same data and you plug it into the model. Well, guess what? It works, right? Okay? If you allow me to start with 10,000 variables and let me do fancy kinds of curve fitting, I will always be able to find a predictor that perfectly fits the data. So plugging the same data into the model that you used to develop the model naively is a useless thing to do, because it'll always look like you have a great predictor when in fact you might have a completely useless predictor. So conceptually, this is what I'm talking about. Suppose that the true relationship between this biomarker and some risk outcome is that it's that purple line, right? But if the green X's there are my actual data points, and there's some noise in the measurement of those X values, then if I allow you to fit a very complex model, you can make sure that model runs through every single one of those X's perfectly. But now, if I hand you a new data set that doesn't have the same noise as the first data set, knowing that the true relationship actually is that straight line, then this curvy model that I came up with on the first data set is not going to work well in the second data set. So that's the idea of overfitting. Here's just an example. This actually came out of a very good research team. There was just, I think, a miscommunication maybe between their computational folks and the other authors on the paper, that they did very nice analysis, came up with the predictor, but at the very last step, they slipped up and they did resubstitution, okay? So what you see there is this 15 gene signature in lung cancer was to predict which patients were at low risk for disease-specific survival versus high risk for disease-specific survival. Now, any of the statisticians in the room are probably thinking, you know, when I see a separation in the curves that big, hazard ratio 15, my antenna go up, all right? So actually, some of us read this paper and we were interested in pursuing it. What was really nice about this paper is they made all the data available. They made the computer script available. So we could actually go back and do a reanalysis. And here's what we got when we did the reanalysis. So on the left there is the original picture. Now, one of the problems we faced is that the code was made available, I think it was MATLAB, and the version of MATLAB was no longer available. So we couldn't actually run their code. We had to try and rewrite their code in R, and we got pretty darn close. So of the gray pictures there on the right-hand side, the first one is our attempt to reproduce exactly what they did, and we got pretty close. Okay, so in fact, we actually got a slightly bigger hazard ratio, but that one is using resubstitution. When we used cross-validation, which is, you know, we took parts of the data out, built the model, made the predictions, and then we swapped around, as Giovanni talked some about that. And as he mentioned, internal cross-validation is never as good as an independent data set, because you kind of could have some similar kind of biases in the whole data set. But it's the best you can do if you have only one data set. You need to do some kind of reuse of the data. So you can see there that it's a somewhat different picture. Obviously, the curves are not as far apart now. And clinically, what's probably more significant is that low-risk curve now, I know it's hard to read the numbers, the low-risk curve now kind of levels off a little bit below 75% disease-specific survival. So that has very different clinical implications than the curve on the left, right? Because if you told me I only have maybe 10% chance of having my disease come back or dying, I might be willing to forgo chemotherapy in that situation. This was built on the observation arm of a clinical trial. But now that it's below 75%, I'm saying, well, you know, I don't know if the chemo is going to help me, but I'm not willing to sit around and wait and do nothing if I have a 25% chance of my disease coming back or dying. The other interesting thing about this predictor is that they built this predictor on the observation arm, but what they were really keenly interested in is, could it tell you which patients benefited from chemotherapy? So they took the resubstitution estimate on the observation arm, they applied their predictor now to the other arm of the trial, which was the patients who got chemotherapy, and said, look, this is fabulous. If you're in the high-risk group, you see on the bottom left there that chemo gives you a much better outcome than no chemo, and low risk, other way around. Unfortunately, those two pictures on the bottom are kind of contaminated by the same overfitting bias as was shown in the top there. So again, we redid the analysis and came up with some corrected versions. You still got a benefit in the high-risk group there on the right-hand side. In the low-risk group, we weren't sure quite what's going on. The curves became pretty much on top of one another. If anything, maybe they flipped around the other direction, but not big enough sample size to really conclude anything. Okay, another key point is the idea of validation. And this was brought up already by previous speakers. So one of the things in this data set, again, which it was a nice paper, they said, well, we had some independent data sets. We applied the predictor to the independent data sets, and it validated. Well, what does validated here mean? Okay, the curves are not as wildly separated as they were in the original overfit story. And the hazard ratios, instead of being 15, are now like in the 2 to 3 range. Furthermore, the endpoint has changed, the time scale has changed. There's a different mixture of disease stages. So I don't know what to make of this validation. I mean, I commend them for trying to do it, but it doesn't really get me where I need to be in terms of, is this ready to actually use in a clinical trial? I'll skip that one for sake of time. And one of the previous speakers already gave a very similar list to what I'm going to give here about what do we mean by a validation. Means you have to have everything locked down, a pre-specified performance metric. Validation is not a matter of just getting a significant p-value. You have to show that kind of the conclusions you would draw clinically are the same in your validation as what they were from your original development set. Ideally, this should be an independent validation set when you have it. And you need to have all the other parts locked down. Blinding is always great to have to keep everybody honest and resist the temptation to make changes to the data or the predictor. And I just want to leave with the fact, since I gave you some examples of things that kind of didn't work, that we are now getting some of these genomics predictors that actually do work. And one of the biggest success stories in oncology is the Oncotype DX breast cancer score, and they showed its predictive ability, prognostic ability, as well as its predictive ability. And the pivotal clinical trial that was sort of the final validation of that model came out and showed that, in fact, the classifier does have good predictive value. It helps you choose or identify which women would benefit from chemotherapy added to endocrine therapy in breast cancer. So this is actually a really major advance that we're going to spare a lot of women who have hormone receptor positive disease from getting chemotherapy that will not benefit them and will only cause them toxicity. So I think that there is a lot of really important things that can be done with omics. And we just have to make sure that we continue to educate people on how to do things correctly. We need to make sure that we have the resources that people need in terms of data, and that we have the right kind of expertise involved in these teams.
Thanks very much, Lisa.